In this video, I will explain the basic structure of a neuron and the way in which a neuron functions. All of the elements involved will be portrayed by elements that enhance a woman's natural beauty. The dendrites, the bushy fibers at one end of the cell that receive messages and send them to the rest of the cell, are displayed here as bobby pins. The cell body, or soma, is the head of the brush. The brush handle represents the axon, which carries messages along the cell, with the place that the head meets the handle functioning as the axon hillock, here. At the end of the axon, the terminal branches are displayed as hair clips. These terminal branches end in synapses, which is where one neuron forms junctions with another, although they are separated by a synaptic gap less than a millionth of an inch wide, displayed here. The synapse is represented here as the hair clips almost touch the bobby pins of the next neuron. Along the axon, the thick black hair ties represent the Schwann cells that make up the myelin sheath, which is a fatty layer of tissue that insulates and protects the neurons and also helps to expedite the process of the sending of a signal. The places where these individual Schwann cells meet are called the nodes of Ranvier, and this is where the actual signaling takes place through action potential, which will be explained in a moment. These nodes are symbolically represented by the small orange elastics. Signals travel down the neuron from the dendrites to the axon terminal, which then influence the neighboring neurons to fire. So signals flow in this direction. Zooming in on the cell membrane, represented here by a variety of eyeliners, we can see that the ions on either side of the membrane are in different concentrations. The green sparkly earrings represent sodium ions, while the black earrings represent potassium ions. This gradient is maintained by sodium-potassium pumps, displayed here as an eyelash curler, which are proteins that continually pump 3 sodium ions out of the cell per every 2 potassium ions it pumps in. This difference in concentrations of ions makes the resting potential, which is the difference in electrical charge across the membrane caused by these different ions. The inside of the cell becomes negatively charged, while the outside becomes positively charged. When a cell is excited by neurotransmitters, it begins to open the selectively permeable ion channels, seen here as tweezers, which allows sodium ions to rush into the cell. This makes the inside of the cell become positive and thus signals the pump further down the axon to do the same thing. The aforementioned nodes of Ranvier are the places where this depolarization takes place. In order to repolarize the membrane so that the neuron can fire again, the cell utilizes the sodium-potassium pump. Zooming back out, we see that once the change in charge reaches the end of the axon terminal, it stimulates the release of neurotransmitters, displayed here as cotton balls, which cross the synaptic cleft to bind with protein receptors on the dendrites of a neighboring cell. Depending on the type of neurotransmitter, it can either be excitatory, or encouraging the next neuron to fire, or inhibitory, discouraging the next neuron from firing. When the excitatory signals outweigh the inhibitory signals, the next neuron will fire, and the whole process will begin again. Upon completion, the neuron that sent the signal absorbs the neurotransmitters back into the axon terminals in a process called reuptake. And here's a kitten acting as multiple sclerosis as she attempts to destroy the Schwann cells.